Good afternoon, all. Is my voice clear and audible to all of you? Can all of you hear me clearly? Hello? Can you hear me? Okay. So I hope all of you are doing well. Maybe all of you have already vaccinated. So today we will be going to start our semester 2 with the subject immunology. Now, do you have any, I hope you have already have the basics of immune system, like what are T cells, what are B cells, even though in the slides I have included all the basic things that you need to understand the concept of our immune system, which will help you to understand the drugs which are used when we'll discuss about the drugs in our scientific basis of therapeutics too. So basically in this immunology class, we'll be discussing the basics, only those basics which will help you to understand the concept, and that concept which we will apply in your career in deciding the drugs, and also to avoid the drug to drug interaction in pharmacology. So I hope most of you have already joined. So, okay. After this class, I will upload the notes in your portal. Also, this time what we will do, I will upload in advance all the notes that we'll be discussing in our class. That before class, also have a look into the notes. So if you have any questions, anything, as usual, you may ask, or you can also mention in the chat box. Okay. So let me share the screen first. Still a few people are joining in, so let them join. We'll start in 1 minute. Okay, suppose during the lecture session, if you have any doubts or if the net is fluctuating, just mention in the chat box. Okay, if you are facing any problem, you can mention in the chat box. Okay, so this is our lecture outline for today's session. We'll discuss about the basics of immunity, the immune system, what is immune response, the definition of immunology and the basics, also the defense against infections, that is the first line of defense, second line of defense, as well as the third line of defense. Okay. Also, we'll discuss about what is the difference between innate immunity and adaptive immunity. Okay. Okay. Is there any class representative for this class 2? If anyone would like to nominate yourself as the class representative, so that it will be easy for us to communicate between ourselves regarding quiz, regarding assignments. Okay. So let's start with the first topic, that is the terminology, to understand the terminology of immunity and immune system. as well as other terminologies which are associated with our immune system. Now, first of all, if you speak about immunity, what is immunity? Right. In simple English, if you say immunity means a type of security, a type of resistance. So immunity here means resistance to a disease, but specifically infection or the disease which can cause infections or some infectious disease which may be induced by an individual by an infection or vaccination. That is, we call it active immunity, or it may be induced in an individual by transfer of antibodies or lymphocytes from an actively immunized individual. We call it passive immunity. So basically, immunity means it is a resistance to a disease, especially the infectious disease, which can be caused by bacteria and viruses. Okay. Now, let's talk about immune system. Now, immune system means here you can say the collection of all the cells, tissues, molecules that mediate the resistance to infection. Okay. Now we have different types of cells as well as tissues and also molecules that provides the defense mechanism of our body, which can resist the infection to cause in our body, such as to protect the body from harmful effects of infections or some foreign substances, also to destroy the abnormal cell, which can give rise to cancer cells. Further, also to prevent the abnormal breakdown of the body's own tissues. So these are some of the function of our immune system in brief, but we will study in details what are the function of our immune system. Suppose if we speak about immune response, how to explain a immune response? Now this immune response is coordinated reaction of the cells and molecules to infectious microbes. That means when any foreign substance, right? When any foreign substance invade our body, that means goes inside our body, right? So our body will initiate a response. That response is called an immune response. Okay, and which is coordinated by cells and molecules that is present in our body. Now, immunology means, as we know, immuno means immune system and logy means study. So immunology means the study of how the body protects itself against the invading pathogens and harmful cells or any substances. So here we'll study about the mechanism of action of the tissues, cells, and the mediators, how they initiate the immune response. Or in simple English, you can say how they will fight against the foreign bodies such as bacteria and viruses. Okay. Now here you can see the components of the immune system. And also you can see the foreign bodies which are involved or which can initiate the immune system, such as bacteria, parasites, pollution, toxins, fungi, and viruses. So what they'll do, they will act as an antigen to our body, which will give rise to the activation of immune system. Is it clear now? Till now, anybody has any doubts? Anything you like to ask? Now, this is the importance of the immune system. First of all, the role of the immune system and their implications. Such as first function of this immune system is to defend our body against infection. Okay. Now, implication means what will happen if the defense system against infection is not working. So here, this is the role and this is the implications. Okay. So in implications, you can see that if there is a deficiency in the immune system, what it will be resulted into? It will be resulted in increased susceptibility to infections such as AIDS. So vaccination boosts immune defenses and as well as protect us from infection. Secondly, the immune system will recognize and it will respond to tissue grafts and newly introduced proteins. Now, this is generally regarding tissue transplantation or organ transplantation. So when we transplant one organ from a donor to a receiver, sometimes what happens, the receiver body will not accept it. It's rejected because of the immune system. Okay, because these immune responses, they are very important barriers to transplantation and gene therapy. Okay, so here you can see that the immune system recognizes and responds to tissue grafts, that is organ transplantation. So here in implications, immune responses are important barriers to transplantation and gene therapy. Because suppose you take uh, suppose you take a country. So if you want to visit a country, you need to have a valid visa. Then only you can enter. Correct. So without visa, if you enter, what will happen? You will be arrested. Correct. So during organ transplantation, what will give? Will give immunosuppressant, so that the receiver body will accept the organ. Okay. Now, third is the defense against tumors. The immune system also plays an important role in defending our body to form tumors. That means it also acts as an anti-cancer element. And that is also very, very potential for the immunotherapy of cancer. Then, fourthly, antibodies, they are highly specific reagents for detecting any class of molecules. So, in case of implications, immunologic approaches for laboratory testings are widely used in clinical medicine as well as in terms of research. Okay, anybody has vaccinated? Anyone? Anyone of you have vaccinated? So
Yes, yes, for the COVID injection, you have explained that for the COVID.
yeah, sorry, I can't hear you properly. Can you write in the chat box? Okay, as Suchira, right? Suchira said yes. Okay, so which vaccine did you take? Or Mr. Janaka, as mentioned, yes. So which vaccine did you take? Is it from Pfizer, from AstraZeneca?
AstraZeneca, sir.
AstraZeneca, okay. So do you know this AstraZeneca, what type of vaccine is it? Is it DNA? Is it a protein? Okay, all health workers vaccinated AstraZeneca. Okay, that's good. That's good. Okay, Sinopharm. So you know what does this Sinopharm contain? So what is the difference between this AstraZeneca vaccine and Sinopharm? So at the end of this today's session, possibly you may be able to understand, because this is very, very important and very, very complex also. Okay, I will explain it. I will explain what is the difference between this Sinopharm. Sinopharm, it is from China, right? Yes.
Those vaccinations produced by the using genetic technology. It is
Okay.
different.
Yes, it's different. How is different?
Some are produced by using the DNA, some are for using the RNA for the
Yes,
technology.
yes, correct. Very good, very good. So what happens? Traditional vaccination, right? Traditional vaccination, what they contain? They contain the weakened or the killed virus, right? Traditional way, like the vaccines from Sinovac or Sinopharm. But the vaccines from Pfizer's, AstraZeneca, what they did, they formulate the protein, right? They formulate the protein from the virus, not the virus, right? And they, when that protein will be injected in our body will produce the antibodies. So the outcome is same, but the process of initiation is different. So if you search some good websites or authorized websites, you can find that efficacy of different vaccines are different. Suppose for the Pfizer, the efficacy is around more than 85%, whereas the efficacy of AstraZeneca is around 70 to 78%, whereas the vaccines from China, they are around 50 to 60% only. So that's why nowadays we can produce the vaccines by inducing the protein factor that is actually causing the we can say the pathogenic factor in the body, which will give rise to antibodies. This one we'll discuss in our coming classes also, which will be easy for you to understand. But as it is an introductory class, I am just giving you the we can say the basics. Okay. Okay. Now here, now if we speak about immunity, right? So as a whole, we call it as a defense mechanism in our body, or the defense system in our body. So when we speak about our cells, our tissues, we call it as internal defense. But we also have some external defense, such as the lysosomes in tears and other secretions. Now, okay, do you remember what is lysosome? Anybody? We have already studied in our human anatomy and physiology class, lysosomes. Anyone? If you want to answer, you can answer in the chat box. What are lysosomes? Oh no, I cannot say antibacterial, right? Lysosomes cannot say antibacterial. You can say lysosomes are enzymes. Yes, organelle. Correct. Lysosomes is an organelle, right, which contains enzymes which will destroy certain cells. Okay, so as these lysosomes are also present in our tears, so when it will come in contact with the bacteria, it will kill the bacteria. Yes, enzyme store unit, correct. Okay, so this is also a type of external defense. Now also you can see here removal of particles by rapid passage of air over turbine bones. So here we have hairs, here we have some bones, but they will restrict the passage of air which may contain some particles. That is external defense. Also, you can see here mucus, acid. Now, generally nowadays we are not eating outside due to this COVID. But before COVID, I hope or I think many of you have eaten outside from the outside vendors, right? So when you eat something from the outside selling vendors, like vada or anything that may contain some bacteria, but generally when you eat it, we never get fall sick so easily, because when the bacteria goes inside our stomach, it will be killed by the acid. Okay, so that is, you can say, more towards physical interactions with the bacteria. So all these are the exterior defense mechanisms, but we'll be more focused on immunology. When you speak about immunology, we'll be more focusing on our cells and tissues. Like here, like here you can see tonsils, lymph nodes, tonsils, lymph nodes. Here also you can see the lymph nodes where they are present. Thymus, generally thymus, we don't have thymus now. Until the age of 12 or 13, we are supposed to have the thymus gland, which will train the T cells and B cells to execute their immune response in our body for the rest of the life. Okay, now tell me one thing. I hope you have frequently visit doctor, right? So when you visit doctor and you say that you are having some fever, the doctor always touch behind your neck and he or she tries to feel something. What is that? Just below our neck here, the doctor tries to press here and try to feel something. That is the lymph nodes. They try to feel whether the lymph nodes are inflamed or not. If the lymph nodes are inflamed, that means if the size of the lymph nodes are increased, that means that person is suffering from some infection. And why the lymph nodes have increased in size? Because they are producing more cells, more cells which will be fighting against the infections. That's why the lymph nodes becomes bigger in size. And that's why the doctor always touch just below the neck to feel the size of the lymph node. Okay? You're also seeing. Now here you can see primary immune surveillance and secondary immune surveillance and tertiary immune surveillance. So primary immune surveillance means, here you can see when this one, let me, okay, here you can see this is the lymph node. So when any foreign substance will be invading in our body, right, this lymph node will be activated. Right, this lymph nodes will be activated and
Hari,
it will
nah, emang
generate.
wajah keluar perlawanan charge kerana manggilan tenang.
Okay. So these T cells now will recirculate through the local lymph node seeking for the antigen. So when there will be antigen present in the body, the lymph nodes will be activated and it will go and bind to the antigen to destroy it. That is the primary immune surveillance. In case of secondary immune surveillance, what happens? Here antigen, which is non-specific, right? Non-specific requirement of CLA plus effector cells or memory T cells. Now here what happens once our body is infected, right? Once our body is infected, our body will produce the antibodies, but our body also remembers the antigen. So what happens once a person has suffered from chicken pox, the person doesn't generally the person doesn't suffer from chicken pox again in his rest of his life, because we have certain T cells which we call as memory T cells. So they remember the type of antigen and quickly they will produce the antibodies to defend our body against the infection. In case of tertiary immune surveillance, here we have the lymph nodes. Also, we have the other cells like central memory cells, T cells. They will recirculate and they will seek for the antigens to remove it from the body. Okay. Now here you can see, you maybe get confused that where are these cells are present. Either these cells are present in our tissues or these cells are present in our blood. So in blood, what are the defense mechanisms that we have is neutrophil, eosinophils, basophils, and monocytes. So these are the four components that we can find in our blood. In tissues, we can find macrophage as well as dendritic cells. But this macrophage, they are formed from the differentiation of monocytes. That means monocytes will undergo differentiation and will give rise to macrophages. Now lymphocytes like B cells and T cells, they are present both in the tissues as well as in the blood. Now comes the actual part of today's lecture session, that is defense against infection. How our body defends against the infection. So generally it consists of two types, innate immunity and adaptive immunity. Innate immunity means it's natural, it's native, and it is non-specific immunity. That means innate immunity is present from our birth, that we may have from our parents. Whereas adaptive immunity means it's very, very specific or acquired immunity. That means we have acquired that immunity through some infection or through vaccination to a specific kind of infection. But innate immunity is non-specific. Now, I hope you may have heard some cases that due to COVID, some people they have suffered drastically and they have died. But also you may heard cases that some people they have suffered from COVID, but nothing happened. They stay at their home, they eat well, they do all such type of things that they do when they was in normal condition, and nothing happened to them. After 14 days or even after 7 days, they were you can say they were cured by COVID. No medication required. So that is because of the innate immunity. Now, what are the defense mechanism of our body? The classifications of the defense mechanisms of our body. Now here you can see this is the host defenses. So what are the host defenses? Host means us. Now we have innate immunity and we have acquired immunity. Okay. Now in case of innate immunity, we have again two types. First line, second line. Now first line means the physical barriers like our skin, our gastric acids, our secretions, and also some genetic barriers. That means a person who is genetically immune to certain disease. Okay. Now the first line of defense is a surface protection only, which depends on the anatomical as well as physiological barriers that will keep the microbes away from the body, or they'll keep the microbes restricted to enter our body. In case of second line of defense, you can see we have inflammatory response. We have interferons, phagocytosis, and the complement system or the complement factor, that is major histocompatibility factor. Now this is basically cell mediated, or we can call it internal defense. Okay, so here we have some cellular response as well as chemical response that will immediately come into play if the infection agents enters our body. For example, such as phagocytes. Phagocytes, what they will do? They will engulf the foreign matter and it will destroy it. An inflammation which holds infections in check. Okay, I'll give you a simple example here. Suppose this is our, we can say this is our tissue. Okay, some insects bites here. When some insects bites here, what will happen? This area will become red in color. This area will become swollen, right? So this area will become a bit warm. So these are some of the symptoms of inflammation. Why it will happen? Because when the insect bites, right, here our body will take the vector of the insect as a antigen and will activate the immune response through these inflammatory mediators such as histamine, cytokines, then tumor necrosis factors. All those things will be activated. So in case of acquired immunity, now this acquired immunity can be natural. Also, it can be artificial. Okay, anybody can explain how we can acquire naturally immune? How we can be naturally immune? Or how we can acquire immunity against a particular disease naturally? Okay, very good. Yes, from mother's milk. Yes, correct. And any other way except from that? From antibody? No, naturally. Antibody means artificially acquired. That means we are taking antibody from some other person and then we inject in our body. You remember that is the passive immunity. Okay, vaccine life attenuated. This is also comes under artificially acquired.
Infection.
Yes, by infections. Thank you for answer. By infection. Now suppose Now, suppose a patient is infected, right? A patient is infected from chickenpox, naturally infected. So, chickenpox, as you know, chickenpox after 21 days, 21 or 28, 28, I think. Generally, after 28 days, the person will be totally cured, right? So, what will happen that time when the virus will leave the body of the person? By that time, that person's body or that person's immune system will develop the antibodies. So, that is naturally, right, without any induction. So, infection, maternal antibodies, as we have already answered there. Okay, so these are the two ways how we can acquire natural immune defense. Artificially means through vaccination, through active vaccination, or through passive vaccination. Active vaccination means we give the weakened or the dead virus, and passive immunity means directly we are giving the antibodies. Okay. Now, in this case, you may also heard, right, that some people after giving AstraZeneca, they are having pain in their hand, they are having high fever, vomiting, diarrhea, and so on. Also, you may have that some people that doesn't have any response, means no fever, no pain, even though there are rarest of the rare case, that is blood clots. So actually, this blood clot actually is this related to the vaccine. Is there any cases in Sri Lanka regarding blood clots after giving AstraZeneca? So these are some of the effects. Suppose here also you can see the third line of defense, which includes specific hosts. Yes. Which one, Mr. Janaka? Yes. Okay. Okay. Now, if you go through some journals or if you go through some scientific articles, right? Although is the rarest of the rare case regarding the blood clots, some people say that it happens due to the person who injects the virus. Means there is a something wrong in the administration process. That means when you inject something directly in our artery, it will clot blood. But actually, vaccines we are taking where? The vaccine should be given in our muscles. So that is one of the factors, as per some scientific articles and some senior doctors. But still, we don't have the correct answer to that. Why AstraZeneca vaccination caused blood clots in 1 out of 1 million people? Okay, now this one, the third line of defense, you can see here, host defense, then developed uniquely each microbe through the action of specialized white blood cells. And this form of immunity is usually long term and has memory. So, especially you can see here that the acquired immunity when you give vaccination, right? Suppose in our childhood, we have been vaccinated for at least some 6 to 7 types of vaccination, so that for the rest of the life we don't have to worry about that particular disease. So this comes under our third line of defense. Now, this is the difference between innate immunity and adaptive immunity. Innate immunity is very rapid, whereas adaptive immunity is slow. It will take some time to develop the antibodies. Now, what are the cells that are involved in the innate immunity? Yes, anybody would like to ask anything? Oh, okay. Now here you can see the components of innate immunity, such as the macrophages, dendritic cells, right? Then natural killer cells, basophils, eosinophils, neutrophils, mast cells, which contains histamine. So these are the innate immunity, which responds within, you can say, few minutes. Just like, as I have explained before, just like when an insect bite, the first thing that will be activated is either, you can say, neutrophils or either histamine. So allergic response, right? Allergic response is also a type of immune defense. That's why all people are not allergic to seafoods. Now, in case of adaptive immunity, here you can see that it is composed of B cells and T cells, especially. Generally, CD4 plus cells and CD8 plus T cells. But in case of both the immunity, these two cells are, that is, gamma delta T cells and natural killer T cells. They are common in both the immunity. Now, this is an overview of the body's defense that we have discussed. Now, this is we can say this is a type of summary. Now, first line of defense, it is intact skin, mucous membranes, secretion of skin and mucus, and normal microbiota. Whereas in case of second line of defense, you can see here, it is generally composed of natural killer cells and phagocytic white blood cells, inflammation, fever, antimicrobial substances. Whereas the third line of defense is specialized lymphocytes, that is T cells and B cells antibodies. Now you can see here the third line of defense is only activated through adaptive immunity. You need to remember this type of things which may arise in your exam questions. That is, the third line of defense is only established through adaptive immunity. Okay. Now here also you can see the timeline, means how quick they work, like innate immunity and adaptive immunity. Now the innate immunity, they provide initial defense against the infection by activating the neutrophils or by activating the macrophages. But in case of adaptive immunity, it needs to develop. It needs to develop the antibodies. That's why it takes time. Okay, do you know after taking the first dose, how many days it needs to develop the antibodies in our body regarding this COVID? Anybody would like to share your views? After taking the first dose, how many days it needs to develop the antibodies in our body? Okay, 14 days. 14 days, I got an answer, 14 days. Others, do you agree with 14 days or do you have any different opinion? So you have, all of you have taken the second dose too? So if we have taken the second dose, after how many days you have taken the second dose? After 1 month, you have, oh, it's too quick.
After 2 months, sir.
After 2 months means after 8 weeks, right? After 1 month means it's too quick. Why it is too quick? Because our body. Okay, let me make it clear so all of you will not have any doubts. Okay, when you take the vaccine, right, it's an example of acquired immunity. Right? So it takes time for our body to develop the antibody. So day one, when you take the vaccine, after generally 21 to 28 days required by our body to develop the antibody. That is almost 1 month. Correct. So when the body will develops antibody, right? When the body develops antibody, our body becomes very weak. Why it becomes very weak? Because our body is working a lot to develop those antibodies. So at that period of time is very crucial because we may be infected by some other infections other than COVID, right? Because our body is functioning towards preparing antibodies to a specific type of disease. That's why there is a buffer. We call it buffer timing from the first dose to the second dose. So the buffer time is around 3 months. That means within these 3 months, we need to take the second dose. Okay, so that's why the schedule is, you can say, 6 weeks. Okay, now understood all of you? Do you have any questions to ask? Okay. Now here you can see that why it takes time. So the B lymphocytes, what it will do, it will produce antibodies, but during the production of antibodies, there are many sub-compounds. Same goes for the T lymphocytes. Now, introduction to innate immunity, that is our first line of defense as well as the second line of defense. Now you can understand that present at birth. Right? This point all of you must have understood. It is present at the birth. So always present and available to provide rapid responses to protect us from disease. Also, it will act against all the microbes in the same way and does not involve any specific recognition of a microbe. That means whether it is a gram-positive bacteria or gram-negative bacteria, or whether it is a virus A or virus B type, it will act in a we can say same way. The mechanism of action is same, but it does not have a memory component, and it cannot recall a previous contact with a foreign molecule. So here you can see the difference. It doesn't have any memory cells. So it cannot recall a previous contact with a foreign molecule. Now it represents immunity's early warning system. That means fever. So generally, if you take vaccination or if you are exposed to some infection, our body will initiate the immune response, which may increase the body's temperature. Now it is designed to prevent microbes from gaining access into the body. Also to help eliminate those microbes that do gain access into the body with the help of phagocytosis. Whereas in case of third line of defense, that is the acquired immunity, it involves specific recognition of a microbe. That means specific vaccine to specific infections. And it is slower in response because it needs to produce the antibodies in our body. And also it has a memory component. It also involves lymphocytes, which is called as T lymphocytes as well as B lymphocytes, and their products such as antibodies. Now, this type of defense mechanisms, they are specialized to combat different type of infections because our body will produce antibodies respective to the infections. Also, some activated T lymphocytes can eliminate microbes which are living inside the cells. Now, we'll discuss in detail about the first line of defense. Now, regarding the first line of defense, it is non-specific innate immune system, physical barriers, chemical barriers. Now, here, if you go line by line, you can see that this is the summary of what we have discussed just now. Okay, so you can just go through it. Here also, the same thing regarding the second line of defense, such as it will give a early warning of the infection, fever, inflammation, phagocytosis, and these are all the mediators you can see. Suppose eosinophils, basophils, neutrophils, macrophages, they are all the mediators of immune response in terms of cellular mediators. But in terms of chemical mediators, we have interleukins 1, interferons, and complementary system. Now here you can see that the innate immunity or the innate defense, how it works. Okay, here you can see very simple. The microbe, when it adheres to the phagocytes, it will engulf the microorganism. How fever acts as a defense. Okay, I'll explain it now. Now, fever, actually, fever doesn't act as a defense. Fever is a symptom that our body is responding to some infection, or our body is producing the defense mechanism against an infection. So what happens generally when our body is exposed, right? When our generally when our body is, okay. Now, as per your question, you asked how fever acts as an infection defense. Fever is not acts as a defense. Fever is a symptom that our body is acting as a defense. Now, how? Now, when our body is exposed to some microbes, right? Let's say any virus, any bacteria, or any substance. So what our body will do? Our body will take it as a antigen. So when our body will take it as an antigen, it will activate the immune response in our body. So when our immune response is activated, like inflammation, like phagocytosis, then migration of leukocytes, all this process, as a secondary effect, they will increase the body's temperature. So when the body's temperature is increased, not very high, that one you need to look after. That is not very high fever, mild fever. When there is a very mild fever, so we can presume that our body is exposed to some infection. Is it clear now? Means how fever not acts as a symptom. Okay. Okay. Thank you. Here you can see how phagocytosis occurs in the body. The microbes, this is the microbe. So this microbe is being engulfed by the phagocytic cells. So take it as a macrophage. So this macrophage engulfs this antigen inside. So when it goes inside, here we have lysosome. which contains the enzymes for destruction. After that, what will happen? This is the phagocytic vesicle. Still, it is inside the cell, but still it is covered by a membrane so that it doesn't infect the cell itself. So this lysosome will go and it will take over this antigen and see what it does. It will break down into pieces, means destroy, and then it will go out. So when it will go out, it will go out where? It will go out in the blood. So when it will go out in the blood, it will cause inflammation, right? So it will cause inflammation. So again, inflammation is a symptom of, we can say, any infection, early symptoms of any infection in our body. So why there is inflammation? Because of this phagocytosis. But as far as this blue color one is inside the cell, there is no inflammation. So that's why inflammation you can see after 1 or you can say after 24 hours, or sometimes it may occur within 12 hours also. Now, when you speak about adaptive immunity, our third line of defense, it is again of two types: humoral immunity and cell-mediated immunity. Now, generally, whether it is humoral or cell-mediated, they will provide specific defense against infections. They will provide long-term protections with the help of T cells and B cells. So regarding T cells, we have helper T cells, and also we have cytotoxic T cells, which we call as natural killer cells. In case of B cells, we have memory B cells and plasma cells, and also antibodies. Now, when you speak about humoral immunity, humoral immunity is mediated by some proteins, which we call as antibodies, which is produced by B lymphocytes. Again, the important point here, antibodies are produced by B lymphocytes. So those antibodies will be secreted in our systemic circulation, also in our mucosal fluids. And what they'll do, they will go and destroy the antigens. That means they will neutralize and eliminate the microbes, which is present in our blood or that is present in our lumens of our organs or any tissues. Now, in case of cell-mediated immunity, what happens is that the antibodies is to stop the microbes that are present at the mucosal surfaces and in the blood from gaining access to the colonizing host cells and connective tissues. So here, cell-mediated immunity, what they're doing, suppose in case of cell-mediated immunity, the antibodies will be there and they will prevent the infections, or they will prevent the microbes from entering in our deeper tissues, or they will help to stop the spreading of the microorganisms. Now, antibodies do not have access to microbes that live or divide inside the infected cells. Now, suppose there is a cell, right? So inside the cells, their DNA or RNA, they are dividing. That time, these antibodies will not be able to do anything because that thing is inside the cell of the microbes. Now, this type of immunity, they have defense against intracellular microbes because it is mediated by T cell lymphocytes. So cell-mediated immunity is basically mediated by which lymphocytes? T lymphocytes. Now here you can see what happens here. We have the stem cells, and all of you know what is stem cells. This is a basic type of cells that can be developed into a complete body. Now here you can see the stem cells. We have some unknown processing areas due to which the B cells will be produced, and these B cells will activate the lymph node, which will also be activated by the antigens, and that time, under the effect of antigens, these lymph nodes, they will produce the antibodies in our plasma cells. Now, in case of thymus, this thymus will be also activated by the antigens, and it will produce the T lymphocytes, and also those antigens will be activated the lymph nodes, and these lymph nodes, what they will do, they will produce some small sensitized lymphocytes, which are also a T lymphocytes. So those T lymphocytes will go and kill the antigens. So this is the difference between the humoral immunity and cellular immunity. So in case of humoral immunity, B cells are involved, whereas in case of cellular immunity, T cells are involved. Now this is the, you can say, for your reading point of view, or for your studying point of view, I have summarized the function of the different types of immune cells that are present in our body and their respective functions, such as in case of lymphocytes, B lymphocytes, T lymphocytes, and natural killer cells, we have uh, the function of the specific function is recognition of an antigen. B lymphocytes, what they'll do, they will act as a mediator of the humoral immunity, whereas T lymphocytes will act as a mediator of cell-mediated immunity. Now, antigen presenting cells. Antigen presenting cells, what they'll do? Antigen present. This antigen will be presenting. These are the cells which will take the antigen and present to the lymphocytes so that they can kill it. So they will capture the antigens for display to lymphocytes. After that, the lymphocytes will be activated and it will kill the antigen. Then we have some effector cells like T lymphocytes, macrophages, granulocytes. So all these things, what they will do, like they will eliminate the antigens, like T helper cells, cytotoxic T lymphocytes, then macrophages, through the help of phagocytosis, they will remove all the types of antigens. Now, this is a general pathway that I have mentioned here. Now, here you can see whenever, let me draw it here. Okay. Now, whenever this antigen, right, whenever this antigen will come inside the body or will invade the body, what will happen? There is a type of cell, we call it antigen presenting cell. So this antigen presenting cell will take this one, will have a bound. It will cause a binding and it will activate the major histocompatibility factor, MHC2. We call it major histocompatibility factor 2. That means it is a factor which will again activate another type of cell. That is, it will activate the CD4 plus cell. Now, these CD4 plus cells will also activate the T helper cells, as well as the T helper cells also will activate the macrophages, plus it will activate the killer T cells. Further, it will also activate the B cells, and that B cells will cause the production of antibodies. So this is the general summary of the immune system. General summary. So now you can see that if I ask you what is the difference between the T cells and the B cells, in one sentence, anybody can answer. In one sentence, what is the difference between the T cells and B cells in our immune system? Anybody like to answer? The difference between T cells and B cells in producing immune response in our body. Anybody would like to answer? Okay, I have few answers here. B cells produce antibodies. Okay, B cells produce antibodies. Yes. T cells mediate. Very good. Yes, that's the thing I am expecting from all of you. Yes, that's the concept. Thank you for your answers. So here you can see that these T cells, they they just uh, activate the B cell. That means they will mediate. They act as a mediator to produce antibodies by activating the B cells. But here again, you can see that we have certain T cells which we call them as T killer cells. They can also kill the antigens, but they doesn't produce antibodies. So when they will kill and then again they will inactivate it. Okay, that means the one-time effect. They have seen the antigens, T cells will just kill them up. But antibodies, what they'll do? Antibodies, they will prevent us from future infections. Okay, so I hope it is clear to all of you. Anybody has anything to ask? Any doubts? Anybody, if you need any further clarification regarding the process? So here is, you can find that the function of the T lymphocytes, especially the function of the T helper lymphocytes, what they're doing here. As you know, these T helper lymphocytes, what they're doing, they are called as antigen presenting cells. That means they will take up the antigen and present to the B cells, go and kill it. Right. So during this process, they will also produce some inflammatory mediators like cytokines. Right. So as you know, whenever these cytokines will be activated in our body, it will cause inflammation. And also it will cause the activation of the T as well as the B lymphocytes. So the main function of this T helper lymphocytes is to present the antigens to the B lymphocytes. And also it will activate the macrophages, which will cause phagocytosis, again, which will cause inflammation. Now here you can see we have thymocytes. Thymocytes means the cells which are present in the thymus. Okay, so thymus, what they are doing? Actually, thymus at the early age, they will train the B cells and the T cells how to fight against the antigens. So once they have trained, they will release those cells in the blood. And from there, from the blood, those cells will be taken by the lymph nodes. So when there is a requirement, the lymph nodes, they will release the T cells. When they'll be going in the blood, and then they'll go into the tissues. Some cells, they will, after fighting with the antigens, some cells will be there which will recognize the antigens so that they will prevent future infections to that same type of microbes. Okay. So anybody has any questions to ask, any doubts? Or do you have any opinion that you want to share with me? Or do you have any specific questions regarding, although it's very introductory session, it's pretty simple, but our coming session will be a bit complex. But if your introductory session is clear, you don't have any doubts, so it will be easy for you to understand our coming lecture session, because there we will study in detail. Like you may have many questions in your mind, like after vaccination, why I didn't feel anything, no fever, nothing happened. So is really the vaccination working or not? There are so many questions. Maybe there are so many myths about vaccination. Uh, you will not believe that when I was studying in the school, there was a teacher who used to say that we should suffer from at least mild fever at least once in 2 months or once in 3 months to know that our body is active against microbes. So that teacher, what he's trying to say that as we are taking fever as a symptom of early warning that our immune system is active, right? But nowadays it's not always like that. The vaccines that we are using now is highly purified. As it is highly purified, what will happen? Most of its antigenic part is not there. So there is no inflammation, there is no fever, but it will produce the antibodies. So knowledge is there, but the application of that knowledge is a bit different now. That's why there are certain countries like the same vaccine, the same vaccine which we can get in Malaysia, and that vaccine also in India is available. But if you take in India, you may have fever there. But if you take the same vaccine in Malaysia, you will not have any fever because of the type of formulation that is purified and highly purified. Okay. Okay. Thanks for your comment. Okay. What books? Okay. Okay. First, you tell me what type of books you want to read. Do you want to read books which is very in detail, or you want to read those books which is precise? That means which is knowledge based for your exam point of view. I have certain books, e-books, which I will try to upload in your portal. If not, then I will can directly send to you through link, Google link. Right, textbook of immunology. Okay, you want Indian writers, means Asian writers or Western writers, which is uh, which is more comfortable to you all? Okay, I'll send you two books, and I'll send you all two books. Okay, one Western and one Asian. So whichever you like, you can go through. Basically, if you speak about Asian writers, especially Indian writers, they will write in very detailed. They will write in very detailed. Okay, I got a very interesting question here. What possible questions come from this lesson? Okay, I was just supposed to ask all of you regarding the questions. What are the questions that can come from this session? But luckily you asked me before. Okay, let me share it again. Okay, you can see this slide. Questions for 5 marks, right? Questions from 5 marks can ask like this. Suppose, explain the process of phagocytosis. Okay, explain the process of phagocytosis for 5 marks. You can see here 5 points. 1, 2, 3, 4, 5 points. Then if you go again, what is the difference between humoral immunity and cell-mediated immunity for 5 marks, or for even say 3 marks? The difference between humoral immunity and cell-mediated immunity. Okay, now here you can see that for 10 marks, okay, for 10 marks you can say discuss the line, third line of defense. Understood. So those are the possible questions that can arise from this particular topic. So what you supposed to learn from here is that first of all, immunity, immune system already you know. Okay, last one. Okay, just a minute. Last one is that yes, discuss the difference between first line, second line, and third line of defense. This one, this slide especially. This one. Here I have mentioned in a you can say point form, but if I mention discuss, it will be of 10 marks. But if I mention explain, you can just write in points like here in points. Okay. So any other questions? Anything else you want to ask? So all of you clear? Is there any other doubts that you like to ask me regarding the introduction to immune system? So I hope all of you have understood it well. So if you have any other doubts, I will upload the slides now. If you have any other doubts, you can go through the slides. And in our next class, yes, we'll get the slides now. Just give me 5 minutes, I'll upload it now. So if you have you go through the slides, if you have any doubts in our next lecture session, we can discuss those doubts and clarification. So tomorrow we have class at the same time, right, for scientific basis of therapeutics. So I will send you the PPT by today. Maybe after 5 minutes, you can see it is uploaded there. So you can go through, and if you have any doubts, anything, we can discuss in our next class. So till then, stay safe and keep revising your lecture session. Otherwise, you may have forgotten that one. Okay, so see you tomorrow for our basic.
Thank you, sir.
Okay. Thank you all for joining.
Thank you very much, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.